and this is where we're at, and welcome back. And so, I sent out Brightspeed announcement that what we'll do today is we'll cover lifespan as our first topic. We had a snow day a week ago, and so kind of stumbled out of the gate with that. But we'll try to cover it. And so let me just show you this. So what we should do this week is work on your capstone journal. And that's that shared document. You have two of them. One of them is when you're writing, and that's not final writing, but for your thinking and your research and pondering things. You really want to be using that because I will look at your editing, at the end of the semester and be able to see, and I can look at it any time, right, to see if you're contributing, and you got to contribute over time. And yeah, Violetta, we'll just try to sit in a kind of a circle a little bit, just on Mondays to try to, build that more of a discussion format than a lecture format out. And so we'll talk about lifespan today. And so work on your capstone journal. And I've talked to two of you outside of class, in some detail. And that doesn't mean those two students are set on their project. We all talked a little bit about ideas, but we really want to move forward and not necessarily move forward with a dramatic thing of like, this is my topic, I'm locked in, but actually documenting your research into options because you can get graded, you get credit for that in your final grade that you learned, on the way. You learned about stuff, it's new to you, and you're looking around and you're pasting in links and just all kinds of stuff, and ideas that you have. It's like a diary, it's your stuff, but we just have to be documenting it. And some people might say, well, I have a physical notebook that I jot notes down in. That's great. What you need to do is to use that and then take pics and put them into that, frequently put pictures into the journal, because you don't have to do it in the document, you just have to be doing it. And in that, doing that, it's a very good skill for us to be working on, is this ongoing project development. And we're never going to delete stuff, just going to add to it like that, and organize it in a way where you might put it little notes into it about where one topic begins, and then you say and it goes down to here when I decided that's not going to be my capstone project topic, like that, right? And so that's that. You got to be working on that, and I think it's probably a new habit, for you to experience, but get into that habit. Okay, so then we're going to talk about lifespan, and in doing this, you really always want to be thinking, I'm going to turn this into an answer for the exam, like that, because we'll have a question on the midterm or the final about lifespan. The final covers everything, and so. That's in class, that's what you're trying to do, is tackle these topics in a way that you could write about them and write a page of material about what you learned about them. And I'll have those, I won't just say write about lifespan, I'll guide you to some specific topic, but also say, broadly, in the context of lifespan, what about, senescent cell model, like that. And you might be able to write about that some, but you might be like, oh, I could write a lot about lifespan, and I'll try to put that in. Our exams there won't be any BS on them, but you also can bring what you got. So that's that. So we're going to talk about that today. Then on Wednesday, I'm going to talk about the first chapter of this Gilbert textbook that's on electronic reserve. And it's about introducing the first half of our course, which is the natural plasticity that we see in the environment. What are the terms, what are the concepts, what are the principles, what are the famous examples? And so that'll be really helpful, and that means that next Monday, a week from today, that's what we're going to discuss. We're going to discuss the topics in Chapter 1, and so next Monday you have to, before class, have written down at least 5 things that you could bring to discussion a week from now. And they could be questions, and they could be something from another class, or a job or internship experience that you had that's relevant, or something from the chapter that you want to talk about more, either because you like it or because you're confused about it, like that. So that's our flow, right? And that'll be the next topic we do. And then, yeah, the fourth thing, send pics of those things to me, like that. And I don't look at them ahead of time. I'm not looking at them and going, all right, well, Bryce thought of something, Bryce, tell us about that, like that. I don't look at them ahead of time. It's just, to insist that you show that you are preparing to contribute, like that, right? Okay, and so then as you read chapter 1, as you look into your capstone journal topic stuff and your capstone project topic, write stuff down and bring confusion to class. And it's my job to fit it into our class. I might be like, oh yeah, that, actually that's a really good question for our whole class. Or I might say, yeah, and I'll explain that a little bit, but then you and I should definitely talk, okay, like that, all right? So that's how we're gonna roll in this course, all right? So we won't jump into lifespan quite yet. We'll say, does anybody have any questions, issues about anything at all? Like that, confusion, look at the syllabus. One thing I haven't described that's in the syllabus is the way I teach classes, I record them, and if you miss the class, you have to take notes and send me pictures of your notes within a week. Because we gotta move forward, and you gotta just keep up. And it's not okay to just miss and then be like, oh, I'll get notes from somebody, because that's crap. You've experienced that. That's just no good. And this is new learning for all of us, like that, right? It's new stuff, and it builds, and so you really have to keep it on it. And a week is enough time. You could be sick and stuff like that and still manage to look at the videos. So that's why I record and post videos. The main thing is, there's a whole bunch of reasons, but one of them, another one is you might be here and you might just be spacing out, or you might be distracted because you're thinking, and you're welcome to add to your capstone journal. You might get ideas you want to put in your capstone, just go for it. But because of those distractions, in class, I should record it, have that available so you can go back and look at it, at home, or like that, slow it down. You might be confused, watch it again, part of it, find that part, watch it, stuff like that. So yep.
Could we go over just a little bit about our final result, like final product for the capstone project, just to know what we're building towards the journal?
Yeah. So the way to think about our final capstone paper is it's like a term paper. That's big. That's like half of it. It's a term paper, and so you're picking a topic, and then you are learning about the science of that topic. And so you'll have 15 or 20 references that are from real research articles, like that. And so kind of just the same way we do that, and you'd be describing that, giving the background. Some of the articles might be primary research articles. Oh, hi, Madison, right?
Yes.
And you can just pull up, and Mondays we're going to sit in a circle,
yes,
and like that, and talk about stuff. And so, and right now I'm just answering this question Bryce had about the final capstone project paper. So it's like that, and if you are confused at all about what's a term paper like, I just assume you've probably written one from another class. But if you haven't, you're confused about that, let me know. But it's going to be a thing where we're using the scientific literature to really dive in and learn about the details of that topic. You like that? And so that's a chunk of it. And then I haven't started talking about them yet. But we're going to have 3 or 4 like 2-page writing assignments as we go forward that are very different. So one of them, for example, is going to be, we'll have it probably the first one will be write a 2-page paper, like 2 pages, single-spaced paper like that. So like that, about your career interests, your background, your direction, and how it relates to this course and going forward. And that writing is going to be edited and put into the capstone project paper. So the capstone project paper starts out, it's a term paper, but we're going to have these additions to it, and we don't want to say just pile them in. We got to have these, people call it stepped assignments, right? It's too much to do it in one step, jump up onto the top of this building or take the stairs, And so we'll have these smaller writing assignments like that. And we're not just going to tape them, staple them on to a term paper. We're going to try to fit it together like that. And just for example, another short writing assignment is the capstone experience is a general education requirement. And so at some point you took a course that was essentially about ethics as part of the capstone. And so the writing is going to be, what was that? What was that specific capstone requirement? What was that course? And what was it like? And how does it relate to what you're doing now? So trying to build off that experience like that. If you're a transfer student, you haven't fulfilled, probably the exact requirements they have at USM, so you might have, but what's a course like that you would have taken that fits that together? So there'll be these other pieces like that. And then in that context, then the overall writing style in a term paper is almost always a third person. But this capstone project might have a mixture of first-person writing. And in some cases, it might just be all first-person writing. Like, I am looking into this, I found this, I did that. And so, because it really is about, you as an individual student, and not doing this exclusively as an assignment, as a requirement, but rather as an opportunity, and then you're documenting that opportunity. And one way to think about that is, it's like, oh, you already graduated from college, and you're curious about this stuff, and so you're looking into it, like that, and finding out about it. And so what are some things that, you would deal with? And because it's going to have a mixture, too, not of just like the primary research, but you might actually contact a person, an expert, and fit that in. And it might be another professor on campus, like Luke is a grad student, and as your advisor is Rachel, right? Like that. So he might just talk to his advisor, or you might talk to a chemist professor and say, "You've ever heard of any of this stuff?" Lots of times, experts don't know what you're talking about when you talk to them about ecological developmental biology, because it's a new field, and you could talk to them about that. And it's true for also the primary research literature. You'll find articles, and they don't really seem to be writing for our course and for your capstone paper. So you can explain that. You can actually comment on that. You can say, well, it's interesting, because I asked my biochem professor and Pete Woodruff, and he didn't know, so it gets to be more personal writing. Absolutely fine. Whereas back to a term paper, that's bad. You'd be criticized, no, it's not about you. You're supposed to be like a neutral observer. Yeah. Yep. And you'll be doing that, and we'll be putting stuff into that capstone project paper shared document. And I don't think you need to yet. I think it's all about the journal and kind of the note taking now. But pretty soon, we'll have a short writing assignment. Start putting that in there, like that.
So are they all kind of structured a little differently, or?
You mean the little assignments?
No, the actual term paper.
Totally.
Okay.
Yeah, and so I'm not going to give you a rubric for it, because it's not like that. Yeah, it could be very different. Yep.
Kind of just work with you to organize it.
Yeah, and also really work with me to get help and advice, but not because I'm like focused on the product. I've created a product. I'm focused on the experience and trying to support the pathway like that. Yep. There's a quote. It was a title of a book that one of the USM professors wrote once. I guess it's an old quote, and the title of the book was "We Make the Path by Walking." Right. So that's what this is. Right. You're gonna produce this, and so it's really important for me to share these documents. So you get this experience of, you got to be walking, you got to be pursuing it. That's half of half of it is to show that pursuit of it. But even in the final product, you're not expected to turn it into a real sterile kind of scientific writing. It can be personal writing. Yeah, and in that sense, everybody is pretty different. And also the project really different, so some projects will have much more of a science leaning thing to them, and some might have more of a like science, but also lean out into the community like that, and also lean toward your future goals, right? Violetta said she's pretty interested in tropical. I apologize for mentioning Argentina as a possible tropical country. A little too far south, but tropical health and disease. Violet is applying to medical school, right? Like that, so possibly really leaning toward future, maybe even, where you hope to practice medicine, right? And this all ties in with preparation for those kind of things, yeah? Yeah. Yep. And yeah, other questions, so we're just dealing with just general questions for now. Let me see if I have more slides. So again, that we're going to do these things this week, and next Monday we'll be talking about the plasticity, the normal plasticity, and the introductory principles to that, and examples from that first chapter. And there's a couple chapters I'm going to read from that. And so we talked some about those, and I'm just going to skip over those because we talked about them some last time, and that recording is online, if you ever want to check in with it. And we talked a little bit about longevity last time, and so I'll just start by showing this slide again, and saying that I described this thing that people sometimes refer to as insulin, the insulin signaling system, that's basically in all animals, and it's really not about diabetes. It's a more ancestral role that fits into the life cycle. So you can see this is about life cycle and a plan. And a key concept here is, well, as it happens, our species is a species that doesn't seem to have too much apparently doesn't have too much plasticity with regard to lifespan. So I mentioned this last time just to clarify it. So there are two topics here. One is called life expectancy, and one is called lifespan. And life expectancy is something that's affected by the environment. So we live in a country with long life expectancy, and there are other countries that have shorter life expectancy. On the other hand, lifespan is something that's common for all of the people, all the members of our species. And so lifespan would tend to be a little bit longer than life expectancy, because life expectancy is, can we improve the environment to reach that lifespan, like that. And so that's what we're talking about, and it's a evolvable trait. And that's kind of a curious thing, because in biology, we start out thinking about observable traits like pigments as evolvable. But there are other traits that are evolvable. One that's very interesting is instinctive behavior. That's an evolvable trait, right? The organism inherited that behavior from their parents. And so it must be based on a genetic inheritance, and that would be evolvable. And so this trait, lifespan, is evolvable. It could lengthen or shorten depending on the environment, like that. Okay, so that's where we're at. And so now let's just give this a try and see if students have something that they want to share that they ran into in reading about this. I also pulled up our here's that aside writing, but we don't need to limit ourselves to that, right? But you could refer to the aside writing, anything like that. So does somebody have anything they want to ask or share about where we're at?
So after kind of reading this, and I'm probably jumping towards more of the end of the assigned writing with this question, but one of the things that it made me think of, and that I think I learned in the past, is about telomere shortening, and trying to think about how that would fit into this model of aging.
Yep. So at the end of every chromosome, we have linear chromosomes, and in general, all animals and plants have linear chromosomes, so they have ends. In general, prokaryotic organisms have circular chromosomes, so they don't have an issue. And the issue is that just in replicating the DNA, because of the nature of DNA replication, the ends of the chromosomes are challenging to replicate, and as a result, the telomeres tend to shorten after each cycle of replication. The telomeres are just repeated sequences that are added to the end of the chromosomes so that the organism is not deleting important genes. And so we can talk more about that biology of telomeres, but they're there and they're helping protect the important parts of the chromosomes as they get replicated over the years. So a person can measure the length of the chromosomes, and they can use that as a proxy for estimating the age, like that. And older organisms tend to have shorter chromosomes like that. Just as a kind of a side, or kind of a specific example of the significance of telomeres in that apparent aging. So so scientists can take cells from an organism and they can grow them under sterile conditions. So they can provide sterile nutrients and they can grow those in a dish. And they can just keep diluting the cells as they grow and divide and adding new fresh nutrients. And one would think, oh, they're going to live forever. These are cells from me in a dish, and they're not out in the world getting beat up and stuff like that down in the old port and things like that. They're going to live forever, but they don't. And this is the thing that's called the Hayflick limit. And the Hayflick limit is just measuring that. And let's see, why is this not responding yet? I guess I don't have this. Oh, no? Yeah, there we go. So it's just a person's name, Hayflick. And the limit is just that it seems like an individual cell has a built-in finitude, mortality. It's not going to live forever. And you can see that by saying, take some cells from a human baby, putting those in a dish, and they'll divide many times before they kind of fade out and die. On the other hand, if you take cells from an old human, they'll only divide a few times. So in our bodies right now, there is some kind of timekeeper, okay? So that's the setting for normal cells. Then the specific example is, it turns out if you take cancerous cells and put those in a dish, they don't have that limit. And one of the characteristic features of cancer is the cells seem to be immortal. They'll just keep going, and seemingly forever. So that's just it's a curiosity, like telomeres. Luke brought it up. Yeah, they seem to have something to do with aging, and then interestingly, cancer cells seem to be immortal. Okay, so then these other cells, the non-cancerous cells we put in a dish, they're mortal. But you might really want to study them for an extended term. For example, you might have gone out and gotten some from a whale. And you're like, man, that was hard to get these cells. We'd like to keep them and study them forever. And so now people will immortalize cells. They'll try to have a cell that's essentially a fairly normal cell that they can maintain in a dish, but it's not going to die. It's just going to be immortal, And again, under ideal conditions, cells just divide, several times, and then, they're done. And the way that they immortalize cells is to simply insert a gene for the enzyme that lengthens the telomeres. And so there's a gene that codes for a protein, and the gene that codes for a protein called telomerase. And we have that in our cells, and it's used, but it's something that kind of goes down with age. It doesn't, do its job, and then kind of disappears, and so we start losing those telomeres like that. They get shorter. But we can just force it to be constantly expressed by putting in, a modified gene that will just keep making it. And that's usually sufficient to immortalize a cell from kind of just about any species. So yeah, this is all, and that brings up this thing where in many examples, we seem to have just covered aging. There's, we stop it by just doing that, like that, like that. And so there's a scientist I mentioned who really, I think, got this field going before, and she was studying this really simple little model organism. And so I'll just show you a picture maybe of her again, and how she fits in, or at least the organism. So the organism is up there. It's called C. elegans. And this is a picture from your book, but it's out in chapter 8. So, I have some pictures that are from your book that, or not your book, but a book, this Gilbert book. And so that's this early work that was started about the 1980s. And the organism is C. elegans. And so the scientist's name is Cynthia Kenyon, and she was a pioneer in this, very interesting character like that. And so there she is. Okay, so one thing she says about this, she'll say, so, talking about aging, and we have to figure out if that's an example of aging that is a real causal, or is it an effect? And the example she'll say is like gray hair. If we could keep a person's hair from turning gray, would we slow down aging, right? So is it a cause or an effect? And right now, here, we're saying, looks like telomeres are a cause, involved in a cause, like that. At least when we talk about isolated individual cells and see how they're doing. And so that's the story with the telomeres, is it looks like they're very important when we look at individual cells in an ideal environment. And so in that reading, we have the author is doing that. The author is bringing up some of these points like that. And our author doesn't say as clearly as Cynthia Kenyon did. Now, is that a cause or effect? What is it? How are we contributing like that? Okay? So that's some ideas about telomeres and how they fit in. Yeah?
Yeah, I just had a kind of related. There was another example in the reading of sort of another mechanism that appears to be working against us that is built in
Yeah.
and ages us. And it was mostly at the interplay of like the insulin signaling pathway being opposed to FOXO and the kind of rejuvenating effects that having FOXO expressed has.
Yeah.
So I was just curious as to whether triggering that insulin pathway is considered also a cause, and like what kind of benefit is there if we need it to survive? Because you need insulin.
Yep. Yeah. And so that insulin signaling system, when scientists are talking about that, what they're talking about is the idea that animals, and because it's an animal-based story, right? Animals need nutrients to grow, so eat and grow. And you start out simply with that idea of, if you have, like, there's apparently, good evidence now that in Asia, part of the, on average, shorter height of Asian populations has been just a lack of good nutrients. Their diet has been so dependent on rice, and rice is not a very great, broad-based nutrient. So that as better quality, broader nutrients get introduced into Asia, folks are just growing taller, like that, So that would be an example of how you, kind of eat and grow. So we're not limiting it to, grow and get, gain weight, like get obese, but rather just grow like that. And so it's in that. So this is part of environmental, plasticity, right? The nutrients are part of the environment, and they're going to impact the phenotype, like that. Okay, so that's where it's at. And the principle is that insulin is going to, the insulin signaling system, that's going to stimulate growth. So if the nutrients are good, it'll be doing things to stimulate growth. Okay, so that in that context, then it appears that there is not just the simple growth of getting bigger, but also the growing up. So we have to think about that, right? That in our life cycle, we grow as children, and then we grow up during puberty. And there's growth that means getting bigger, like a child. And then there's growing up, which means changing, like that. Now, we wouldn't say those same words if we were talking about insect metamorphosis, but we could probably say the caterpillar grows up into a butterfly. And nutrients are a part of that story too. So it's not as simple as nutrients and grow like a child. It's also nutrients and grow up. And then that growing up in animal populations is very common life cycle is that the animal starts out as a non-reproductive juvenile, and then it goes through sexual maturation and reproduces. And so that's showing that that's a big part of this. See, this reproduction strategy like that is that idea of that the insulin signal also has to do with this decision of timing of when to grow up, so that nutrients are a factor in that as well. Yeah? And can you just kind of repeat again a little bit, Violetta, what you were talking about?
Yeah, no, so that was the first part of it, like what it does, and so it just seemed a little bit almost counterproductive in some ways, because if that is paid by lowering the the life.
Yes.
you
So
know,
then remember. So then in this model organism, C. elegans, that Cindy Kenyon is a geneticist, and so we have these two stories from that species. First of all, it's one of these lab model organisms, so it's one that scientists have learned a lot about. And in general, people will say these organisms, plant or animal, that are like lab model organisms. One of the reasons they didn't really realize it, but one of the reasons that they were picked is because they don't have very much plasticity. They really are going to grow the same. We're going to try to put them under a controlled environment in a lab, but it's a species that already has a readiness to just go, like that, really smoothly like that, right? Because the lab model organism folks were geneticists who were trying to find the connections between genotype and phenotype. So let's keep it simple. That's why they brought it into an environment. And that's one of the reasons, although not conscious, why they picked certain species, because they fit really well with that. So the C. elegans is a little bit of surprise, because if you put it under ideal lab conditions, it just grows up, and it only lives a couple weeks, and it dies, grows right up like that. But it happens in nature and can be recreated in lab, that it also, under certain conditions, goes through a kind of hibernation. And it produces a different, and it does that switch before it's reproductively mature. So as the juvenile, if the conditions are harsh, and the conditions can be crowded or hot, just too hot, or poor nutrients are the, I lost it again here, all right, or poor nutrients. Really importantly, there's evidence of just, my little thing is messing up here. And that doesn't happen very often. Really important as an evidence that this is evolvable is the crowded feature is the animal measures it by a pheromone, a chemical scent that they're all producing. So if it's crowded, you just detect more of it. And if it's crowded, you're like, not good. Too hot, too crowded, poor nutrients. And they go into, they are actually kind of molting. So instead of molt, grow up, they go molt, and they go into a thing that's called a dower larvae. And it has thicker skin, it has these specialized receptors to detect the environment, and it's just gonna wait for 5 months like that. So she starts out with that idea that this is a species that does have a natural plasticity with the rate of aging. And then what she looks for is mutants that don't go into, and let me see if I can get this straight. You have to think about it. We might have to think about it after class 2. Because she looks for mutants that don't go into the dower state. And she starts finding mutants that can live this good nutrient life cycle which is mature and reproduce, but not die. So, you have to repeat again what you said, but I know this is addressing what you said some, and that is, she's finding this ability to genetically modify the, and work with mutants that can live longer without going through the shutdown phase. You can have good nutrients, the signals to grow up, they do grow up, but they just don't fade out in 2 weeks. And they don't last as long as the tower, but she can double their lifespan, which that's significant, right? Going from like 85 years to 170 years is a significant lifespan extension like that, right? So that's what she finds, is she's able to use genetics to start teasing apart these requirements. And the requirements at front is sort of like, if the nutrients are not good, try to delay maturation. If the nutrients are good, accelerate maturation, like that. Yeah. And does that fit into the things you were sort of into what you're asking about? Yeah. Yeah. And so that's an important concept. And also why to think about why do we pick this one as an example, and it's very interesting because there's in the great diversity of life on Earth with all the different species out there, we have some examples that we go, oh my goodness, this organism adjusts its lifespan based on environment. So it's a perfect example of phenotypic plasticity. But then, can we start understanding the pathways and start teasing them apart so it's less dependent or altered like that? Yeah.
So is the purpose of studying the ones that don't necessarily go through this shutdown stage to figure out why they, like what's been mutated to make sure that doesn't happen?
Well, the purpose, I think, I'll say yes, yeah. But I'll mention that, she was in England at this famous Cambridge University in a very famous lab doing this research, and all her attitude the whole time was aging, just looking at aging. And it happened that basically the same time as her, there was a guy in one of the state universities in Missouri who was only involved in agriculture. And he was studying this too, but he was just studying it with the kind of motivation of how could we kill these, because these guys are a soil, a nematode, a soil environment like that, and there are other species of nematodes, not this one, that are crop pests, that will destroy roots of crops. So he was working at it from that angle. So he wasn't thinking about aging. His name was Don. I don't remember his last name, but just like a farmer. I met him several times. He's like a farmer guy and a professor, at a farmer kind of university. And he was just focused on what's the deal with this being able to pause, and trying to figure that out. And he studied the mutants with a different spirit of possible targets for killing them, like that, right? Because if you look at the pathway that this is regulating this, and so I have a slide of that, and this gets pretty complicated fast, but let's just look at a figure I have, and I have to scroll a little bit to find it. So here's a little bit of the figure of what both Don and Cynthia, were studying, and they were competing, and what Don, I think mostly Don gets credit for figuring out. See over on the right, there's this word dafachronic acid, and you see how it's made from cholesterol. So it's showing that all the DAFs are specific genes, and in cell guns they number genes instead of naming like insulin and hemoglobin. They're given numbers. So DAF36 and DAF9 were mutants that affected the ability to go into diapause, and they were needed to go into diet pause. No, they're, I have to think about that. But they're enzymes, and they're enzymes that convert cholesterol to this molecule called dafichronic acid. And dafichronic acid is actually a hormone. So in the little worm, it makes a hormone. And it makes a hormone if the conditions are good, and it will go into this reproductive growth. And so there's a parallel, see, like with us, we have testosterone and estrogen when we're going to grow up. And so that is the, it's not related to those at all, right? This is a very different species. But to grow up, it needs to make a hormone that's called dafichronic acid. And Don was super interested in that because he could probably make a synthetic molecule of it and kill them by forcing them to grow up when the conditions were not good, like that. And they're like, "Whoops, I screwed that up." I grew up and it was crowded and we all died. Like that. So that's what he was thinking about, and so that's part of this story. And you can see there's also this insulin signaling pathway like that. So so then working out these pathways, I don't know, can you just repeat your question too, or your comment, like about?
Yeah, I guess that kind of makes sense. I was just trying to think about like how would that necessarily contribute back to Cynthia's research?
Yeah. So the idea, I think, is that under poor conditions, can we activate the grow up plan? So that's part of it. And then can we activate that grow up plan without getting old quickly?
What does that mean?
Yeah. So it turns out that then we have to look at this, and this it's a very crowded image, but let's see the best. So we can maybe go back to this. Is that we talked about, so here this is showing a cell in an animal, and that's the cell membrane, and so this is the intracellular cytoplasmic environment, and that's extracellular like that. And so here is showing that that there's this insulin signaling system. So there's the insulin signaling factors, and we talked about there are 3 family members: insulin, insulin-like growth factor 1, insulin-like growth factor 2. There's the insulin receptor, and so that's going to be stimulating growth. And then this is the signal transduction pathway, the biochemical pathway to regulate the cell's response to the presence of that hormone. And down there is a protein called FOXO, and FOXO is a transcription factor, and so FOXO is one of those DAF genes. It's an ortholog of one of those, so it's doing the same thing in cell organs that FOXO does in other organisms. And folks have really keyed in on FOXO as being common, right? We don't have dafichronic acid. We don't have the enzymes to make dafichronic acid. We don't have the receptor for dafichronic acid in us. But when you start finding the other mutants, you find, oh, but we have something that's playing the function of FOXO. And so FOXO is a transcription factor, and in the presence of good nutrients and insulin signaling, FOXO gets blocked from doing its job. So FOXO is really important, and its role is in extending the lifespan. So can we get the dafichronic acid pathway activated and stimulate the animal to grow up? Well, at the same time, not blocking FOXO. So can we tease apart those two kind of signaling stories? Like, let's grow up, but we're not gonna grow old. So let's kind of split those apart genetically. And so that's what Cynthia Kenyon was doing. You know, Don was just figuring it out like that. So FOXO is famous, and I say ortholog. It's like, you know, we have hemoglobin in us, hemoglobin in dogs, like that. You can look at the amino acid sequence of FOXO and you're like, this is very closely related. It must be a common ancestral gene, FOXO. So if the conditions are poor, then the insulin signaling system does not block FOXO. FOXO gets to do its job. So we can see FOXO transcription factor go into the nucleus, and it binds to target genes, because that's what transcription factor proteins do, is they bind to target genes, and they either activate them or repress them. And in that simple C. elegans, when the conditions are poor, and it's going to go into the Dower larva, arrested state, that FOXO goes into the nucleus and it activates about 200 different genes. And let me just quickly look through here to see if I have an example of what the kind of general genes are that get activated. Yeah?
In the text, there is something that says, I mean, I'd be quoting the text, it is
Yeah.
possible that FOXO/DAF16 activates the expression of genes involved in producing anti-stress proteins.
Yes. So that's an example of one. And there's just a short list that people will use. I don't have a figure, I guess it's just not in here. Like, what is it? What are some examples of genes that get activated, right? Because scientists are like, oh my gosh, these would be anti-aging genes, right? The conditions are poor, we're going to extend the lifespan, we're going to let FOXO do its job, it's going to activate genes. These genes will be anti-aging genes. And so you said activating some stress response genes, right? Like that. So there are some categories like that. So here, so let me find, sorry, that one again. And so, so another, here's one that, you know, would be a category of genes that are involved in this. So we've all heard that in our diet and health, we are concerned about, we want to get antioxidants. Antioxidants are good. They are good for our health. They'll help us live longer. And the reason antioxidants are good is because they'll bind to free radicals, like that. So we can eat antioxidants. Our body is making free radicals in our normal metabolism, in our mitochondria all the time. And so our body makes some antioxidants. And so some of the target genes for FOXO are involved in increasing the amount of natural free antioxidants. You know, you'd be like, good, like that. To help you see that some, an example in aging is that there are some birds that live some species of birds live a long time. Like there's some parrots, you know, these talking parrots. Some of them live a surprisingly long time. Usually animals that are about the same size, they kind of generally have the kind of similar lifespans. So there's some birds that are kind of, people are like, surprisingly small birds that live a surprisingly long time given their size, like that. And so scientists have studied that, and they've seen that some bird species do this thing where they'll make, even under good environment, plenty of nutrients, they make a surprisingly large number of natural antioxidants, and they actually do a good job putting them in the mitochondria where free radicals get produced during normal metabolism. So let's put them where they're most needed, like that. So people are excited about that. So that would be an example of what does FOXO do. It activates genes that are involved, that the enzymes and stuff in synthesizing and maybe positioning antioxidants, like that, okay? So then here's another one, because it's so the kind of the famous molecules in lifespan are the insulin signaling system, which is the insulin-like molecules, hormones, and then the receptors. And the other two are FOXO and TOR. And I won't write it down, but what's TOR? And it turns out it was discovered in mice. And it's called, I'll write it down, but it's not, you don't need to remember this name, but it's fascinating to hear how people discovered TOR. So there's FOXO, and we should be able to write about, well, transcription factor is what it is, and that means its job is to regulate gene expression, and FOXO gets to do its job under poor conditions. And its job is to activate genes that slow down aging. Like that, right? And so, back to the Cynthia Kenyon's research, she found mutations that would grow up, but would age slowly. So they didn't go into that dour state, that hibernation state, to just be like, let's party, right? Like that. Yeah. She also described this, her research, she says it's like this. It's not like our current medical care, which is try to help an old person hang in there. It's more like you meet somebody, and this is her talking, you meet somebody on a dating app, you go out with them, and they're your age, 35, ready to go play racquetball, stuff, but they're 75, it's slowing aging, like that, okay? Like that. Ash, did you have something?
About two things we've talked about.
Yeah.
So I know that downregulating DAF2, so it makes antioxidant enzymes, but I was wondering maybe if, not just that it's to get rid of reactive oxygen species, but that they're also part of oxidative stress. So maybe it could be that there's more signaling and you need more enzymes to handle that.
Yes.
Have they looked at how much reactive oxygen species are also in the tissues? Is it to handle a higher amount of signaling so there's no damage, or is it just to get rid of reactive oxygen species? I know it's pretty all or nothing.
Yeah, well, and so I don't know, but for the FOXO targets, it has lots. So we could talk about categories like that, right? And so one category that Madison brought up is increasing this readiness to this stress response. Then another category is to increase production of antioxidants or things related to that free radical metabolism issue, and how that is kind of harming the organism as it's living, and so it fits into that overall category. In how we talked in class, we talked about telomeres some. And so the author in the reading brings up these different topics because in turn, each of them has some relevance for aging. And then the researchers are trying to find out, was this like gray hair, an effect of aging, or is this more of a cause of aging? And I would say they're working on those features. And so I don't know. I could also just answer your question, pretty quickly by saying I don't know about that. How much, changes and sort of the how targeted that the production of super antioxidants is, to avoid free radicals. So another category of FOXO targets is related to what this TOR does. And so so we got FOXO, and we want to be able to understand what its normal role is. And then there's this TOR, and just to introduce that is called target of rapamycin. And rapamycin is just, well, why is it called TOR? So it's called TOR in every species, but where it was first discovered was in mice. And it was in studies where they have this rapamycin, and rapamycin is a drug, and it's used in patients that have organ transplant, and it suppresses their immune system. And so the person, that's really common. You used to have a perfect match to get a kidney transplant. You used to have like a twin or something like that. Doctors can now transplant in kidneys that just don't have as good a match. And the way they do that is they have the patient take immunosuppressant drugs for the rest of their life. And one of those is rapamycin. And so, well, we better test that new drug. This was several years ago, new drug. Better test it on mice. So, how does it affect the health of mice? And they found out, well, it extends their lifespan. So, target of rapamycin means that's the protein that rapamycin binds to and affects its function. So it's just interesting to hear, how these things kind of lace together, okay? So then, if we look at this, so TOR's function is normally to increase growth. And so that's going to be this, let's grow up and die. And rapamycin can slow it down like that. And I don't remember, I haven't looked recently to see what's the status on research on whether people who take rapamycin appear to be living longer.
So what's happening in this graph in relationship with Foxo and Tor?
Yep. So we introduced Tor. Tor is involved in growth, like that, growth. And just to tell you this, because it's like, oh my gosh, I can't believe it's this simple. So when we say at a cellular level, let's increase growth, mostly that involves making more ribosomes and increasing ribosomal activity. Not like some detailed little regulation. It's just like ribosomes mean proteins mean go, literally, like that. And so that's part of regulating this aging is that if there aren't good nutrients and you want to slow down, you're going to decrease the number of ribosomes. You're going to decrease TOR, because TOR's job is to crank up growth, and it does that a lot by cranking up number of ribosomes. And then there are certain ribosomal subunit proteins that are like, this is a very good ribosome, it's rolling, like that. So it's just surprising to see that. So that's its function. And so you can see FOXO, when it gets to do its job, it activates genes that block what TOR is trying to do. So it shows it blocking TOR, but it doesn't, I don't think it does that by binding to TOR. I think it blocks its effects. So another set of target genes that FOXO activates are genes that decrease ribosomal activity. Like that. Might even be some ribosomal subunit proteins. They get made, they're part of ribosomes, and they're just not going to make very many proteins like that. So that's showing that relationship. That the arrow means to simulate, and the blockhead means to repress, like that. And yeah.
Like it. How might Cynthia's approach work if that's kind of in that simple manner though? They're both kind of pulling and pushing against each other. Does she look at something different,
well, so then you'd start looking at, you'd start saying, so a geneticist can find mutants that either have a decrease in function or an increase in function, right, like that, right? And so when it's a geneticist, they can do a lot of manipulations of pathways. And when they're doing genetic screens, they're going to find mutants that are mutants in genes that are important in these pathways, like that. And sometimes they're a major thing, like, oh, we found some mutations in the FOXO gene or the TOR gene. In other cases, it might be some of the important sort of secondary targets, like a ribosomal subunit protein that got mutated, and you're like, oh, look at how big an impact this one had, right? Right? I don't know, we could Google it, right? But telomerase is a protein, that means there's a telomerase gene, right? You know, is it targeted by FOXO? We're saying, you know, if you increase telomerase in a dish, right, it's going to increase lifespan. Is that one of the natural targets of FOXO? Then maybe it is directly, or maybe it is indirectly. Does, you know, when FOXO gets activated, do we see an increase in telomerase activity like that? And so Cynthia could find an increased activity, a gain of function mutant allele of FOXO, and see all those changes happen in an organism that is otherwise just happy, you know, a lot of food, a lot of nutrients, not, you know, but yet it's just also going like, yeah, I'm going to put on this pathway. See, so the geneticist and biochemist, you know, but they could start teasing those apart. That goes back, I think, to part of Violetta's early question, like that. Okay? So that's some more information about that. We're doing great. And this is what we're doing. And so there are principles here. Let's see. So we go for like 12:50. Yeah. So there are principles here, and one of them is to kind of try to be able to explain like the big picture, and to divide into some of the details to help illuminate the big picture, like maybe a little bit about telomeres. Something we definitely want to say about, because it's a big deal in this field, and the author writes about it in this chapter, and it's a little bit hard to understand, but there's this senescent cell phenotype that's a big part of aging. And so, we talk down at the level of, inside of a cell, right? What's happening inside of an individual cell. Then there are some more global things, and one of the more global things is that when we have cells that are injured, not functioning well, our immune system eliminates them. So our immune system is surveying both for infections of foreign things, but also cells that are not functioning well. Most famously, our immune system can eliminate cancerous cells. And that's one of the reasons that cancer is considered more often a disease of old age, is because our immune system starts to get weakened. And so, there are childhood cancers, but often we think of cancer as a little bit of an older age disease like that. So our immune system is doing that. And it's not just cancerous cells. There could be cells that are not functioning well, that are older, or that are functioning, just misfunctioning for a variety of reasons. And so those cells are senescent. That means they should be dying. And instead of just relying on them to die, the immune system can help them die, like that. So we have in the senescent cell phenotype two kind of dominant things. One is that cell doing a good job deciding if it should go through apoptosis and just get recycled? And also, is the immune system doing a good job surveying and helping cells that should be out of line? So we start out with the senescent cell phenotype, and then he says, and it's bigger, it's a little microenvironment. And that's a big part of aging. And so we could decrease aging by, dealing with these down at that individual cell level to increase the health of those cells, right? Or we could look at that overall microenvironment and say, can we do a better job clearing that out? And how much is that contributing? How much is that causal, right? So an example that the author puts up, and the author is, talking about the field, the broader field, is what people call chronic inflammation. So chronic inflammation is something that, a person who's getting older, a lot of times will have joint issues and things like that, where they're just chronically inflamed. Some tissues are chronically inflamed. Another example is chronic inflammation in the lungs can be an issue. And when there's chronic inflammation, that can contribute to creating this senescent cell phenotype, this situation where within this tissue we have scattered cells that should be dying. They should be dying and getting recycled, and they're not. And that's going to bring a cause of aging. That's going to increase that aging issue. and then now is that a cause or an effect of aging, right, like that? And our author really likes the idea that that's a big thing, that you can kind of pull, you know, lots of these things together and see them happening in this senescent cell phenotype environment, like that. And so maybe we could decrease aging by providing, say, a drug that would maybe increase the immune system's activity to help decrease that chronic inflammation, decrease our ability to find areas, patches of tissue that is like, wow, that tissue is messed up, and bringing the whole thing down. Yep. So we kind of go from what's the principle here, why are we talking about this, to, you know, what do we know about the nuts and bolts features of this, what are some of the things, the pathways, to how does it fit in, how does it start to fit together? And you'll see that some of the topics we talk about are much simpler than this one. The reason we start out with this one is because it's just so intriguing to think that lifespan would be a topic in the, you know, ecological developmental biology. Right? So that's why, you know, our author wants to talk about it a bunch, and I want to, you know, start our course with it, stuff like that. Yeah.
So like for like an exam question you might write for this chapter, like would it be as open to saying like talking just like what, like how do like senescent cell phenotypes like affect lifespan, or like how does TGF1 or IGF1, you know, affect that? Yes. So it could, you know, like it's really likely to be kind of having two, you know, like one question, but it kind of has two parts, which is, you know, what do we learn about lifespan, to give everybody a chance to write stuff they learned about lifespan, you know, pretty open, you know, like that, because you might have learned, you might dive into this, because I'm going to suggest that you can, in each of these topics, you can, you know, you can, for the required reading, you read it, but then you could just Google it and just start going down a pathway that you're interested in, like how long did, you know, one species of dogs and different breeds of dogs differ in their length of their life. It's an evolvable trait, and it is evolved in dog breeds. Like that. Might even be more open than that.
Yeah. Well, so it gives you an opportunity to bring in that, but then I likely will say, and what about something about, you know, the insulin, and include, you know, description of insulin signaling system, or two or three of those, and you see if somebody knows about, you know, they did a good job, you know, relating, like just, you know, not broadly, but they got into it, you know, the required reading and understood that. So there's, you know, I don't try to, you know, cut somebody short because they spent time on it, but they just didn't go after a specific thing, right? But I also like to find out, like, not just, hey, just talk to me about how broadly, you know, like that.
Kind of the organization of like pretty much all the chapters, like how.
Not the chapters, but our the part of our course goal here is, you know, get a feeling for the principles, and start being able to apply them to other examples and topics like your capstone, but then also try to understand and get to, you know, some of the molecular mechanisms, things like that. Here's just another example, and it's the one I was looking for. So see down at the bottom those gray boxes? Goes back to, I think Madison was sort of asking about, bringing up, you know, there's the stress response that gets activated, so our author talks about that some. And we've talked about some about wondering, I don't know if telomeres are part of the, you know, target, maybe, but definitely antioxidants like that, and growth. So see, we're down here, and good nutrient status can regulate protein synthesis, like that. We talked about that. And then cell death, we talked about that, and also the cell cycle. And so, you know, you get to this thing where you're like, if you zoom out, you're like, oh yeah, that makes sense. But then see, the top is that insulin signaling system, and the bottom is kind of a sensible output, and in between it's just a blizzard of signals. And each of the circles is a protein. That means it has a gene and might have popped up in these screens, you know, as they're manipulating them. And I circled TOR, and FOXO would be on there somewhere. and, oh, they're just to the right of it, FOXO, and they call it FOXO DAV16 because that's what the ortholog is called in C. elegans like that. And so we don't need to dive into this too much, but there are just really famous examples of highly studied proteins. Like over here by those red words, Reaper hidden grim, that's the name for some genes that make proteins in fruit flies that regulate programmed cell death, so that apoptosis, we kind of touched on that a little bit, that's part of the story in this, that it fits into that. And over here on the far left, Salvador, hippo, and warts are proteins that regulate growth in fruit flies. And when I teach developmental biology, we don't talk about growth. We really talk about a different part of development because scientists didn't understand growth very well, but now they understand it really well. And hippo and warts and Salvador do these really interesting things in the normal regulation of growth. So we start out with, our walking around in the world. Oh, yeah, I understand what growth is. And we add to it, that includes growing up, which is different, right, like that. And then, what do we really know about, the biochemical genetic mechanisms of actually regulating growth, like that. And so, we're not expecting you to memorize that at all, but at least have a feeling for what we talked about today, and then in the reading, and then also you can chase down some topics that you're interested in because it might become a capstone topic, and if you start doing that, put it in your capstone journal, or you might chase it down because you're trying to get an answer ready for the exam, and it's not going to be limited to our assigned reading or our class time. So you can show up on an exam, and if I ask a question about lifespan, try to address it. So I'm like, yeah, you learned a lot in our class, and then add more of what you looked into. Yeah.
What if we have an idea for our capstone and sort of what we want to focus on for toxicology, but it's not included in what we've been reading?
That's absolutely fine.
Okay.
Yep, the sky's the limit. A person would say, right, like the advisor or the instructor would say, yeah, so there are kind of two choices that students have for capstone projects, and one is a person might pick a topic from that we're covering in class and kind of kill two birds with one stone. You might love this and want to study it for your whole life, but to get through this course, you might pick something, that we're like, we are learning about it in class. So your capstone project could come up on our exam, yay, Or a person might just be like, that's fine, I got this thing, I'm interested, and I'm going to go after that. So yeah, totally, totally. But we got to talk about it because sometimes projects are like ones I'll be like, yeah, that's not exactly, that won't get you to what we're hoping to cover, like that. Like that, try to give you some help like along that lines, okay? Yeah, and we're, I think, pretty much out of time here. We got like a minute, but we got to start shutting it down. So, right, there's a lot more to say about lifespan. So just like in other classes, right, we got to learn more than what we can manage to cover in the class, and that includes assigned reading, but there won't be tons of assigned reading, You need to read that, study it, understand that for exam, answer questions like that, but also go after these topics on your own and with that same spirit of being like, it's not an assignment, it's just supposed to pique your interest, and you're like, oh, dang it, that's pretty crazy stuff, right? And each of you, I think expressed, in your questions and stuff, you're thinking about this, you're like, what the heck, right? So it's a good topic to start with, right? What the heck, like that, yeah. I'm old enough that I was a grad student when people were really studying this yeast that causes bread to rise, and it's a eukaryotic cell, and there are famous labs like this guy Lenny Garante at MIT, just really famous people. People won the Nobel Prize in Medicine for studying the yeast that causes bread to rise. They were studying things like RNA splicing and cell cycle and stuff like that. Then it faded out, but they study it now, and it's crazy, right? There are humans buying dietary supplements that have been shown in simple model organisms to slow down the rate of aging, including yeast, where people are taking nutrient supplements hoping they'll slow down the human's rate of aging based on what it does to yeast. Like that, you know? So it's fascinating, you know, and lots of relevance for our